This video is Chapter 19.4, Bacteria and Archaea. Key concept: bacteria and archaea are both single-celled prokaryotes. Main ideas: prokaryotes are widespread on Earth. Bacteria and archaea are structurally similar but have different molecular characteristics. And bacteria have various strategies for survival. Humans not only share the environment with prokaryotes, for many species, we are the environment. Up to 500 types of prokaryotes can live in the mouth of a human. In fact, you may have as many as 25 different types in your mouth right now. 1 milliliter of saliva can contain up to 40 million bacterial cells. First, main idea: prokaryotes are widespread on Earth. Prokaryotes, which include bacteria and archaea, are the most widespread and abundant organisms on Earth. Consider that humans are one species with about 7 billion individuals. In contrast, scientists estimate that there are more than 1 billion types of bacteria. That's 10 to the 9th power. And more than 10 to the 30th power individual prokaryotic cells on, above, and under Earth's surface. Bacteria and archaea are an important part of every community they inhabit. These tiny organisms live in just about every habitat on Earth, including the air we breathe. Prokaryotes have been found living inside rocks, in deserts, and in polar ice caps. 1 gram of soil may contain as many as 5 billion bacterial cells from up to 10,000 types of bacteria. Prokaryotes can be grouped based on their need for oxygen. Prokaryotes that cannot live in the presence of oxygen are called obligate anaerobes. An obligate anaerobe is actually poisoned by oxygen. Archaea are prokaryotes that can live in extreme environments. The archaea that produce methane gas are obligate anaerobes. They live in marshes, at the bottom of lakes, and in the digestive tracts of herbivores such as deer, sheep, and cows. These microorganisms release nutrients from plants that animals are unable to digest on their own. In contrast, some prokaryotes need the presence of oxygen in their environment. Organisms that need oxygen in their environment are called obligate aerobes. This group includes several familiar pathogens, such as those that cause the diseases tuberculosis and leprosy. There are also prokaryotes that can survive whether oxygen is present in the environment or not. This type of prokaryote is called a facultative aerobe. Second main idea: bacteria and archaea are structurally similar but have different molecular characteristics. Members of domain bacteria and domain archaea comprise all of Earth's prokaryotes. Domain bacteria is the more diverse and widespread of the two domains, while many archaea are found in Earth's most extreme environments. Some archaea are even able to grow at temperatures greater than 100 degrees Celsius, or 212 degrees Fahrenheit, which is the boiling point of water. Bacteria and archaea have many structural similarities, but important genetic and biochemical differences. Even under the microscope, archaea look very similar to bacteria. For example, both archaea and bacteria are small, single-celled organisms that have cell walls and plasma membranes. Archaea come in many shapes, while the 3 most common forms of bacteria are shown on the screen here. Bacteria are often named based on their shapes: rod-shaped bacteria, spiral-shaped bacteria, and spherical bacteria. Prokaryotes do not have any membrane bound organelles, such as a nucleus containing double stranded DNA. Instead, their DNA is in the form of a circle and is surrounded by cytoplasm. Prokaryotes may also have plasmids. A plasmid is a small piece of genetic material that can replicate separately from the prokaryote's main chromosome. Most prokaryotes can move on their own. Many bacteria and archaea move by gliding or using flagella. A flagellum is a long whip-like structure outside of a cell that is used for movement. The flagella of prokaryotes are attached to the plasma membrane and cell wall. They may be at the end of an organism, or they may have different arrangements over the entire cell. Although similar in appearance, the flagella of bacteria and archaea are structurally different from each other. In addition, their flagella are both structurally different from the flagella of eukaryotes. Many prokaryotes also contain structures called pili that are thinner, shorter, and often more numerous than flagella. Pili help prokaryotes stick to surfaces and to other prokaryotes. A typical prokaryote is pictured on the screen here. It was not until molecular analysis techniques were available that the many differences between bacteria and archaea became clear. Despite their similarities in function and appearance, bacteria and archaea are not closely related. Molecular evidence suggests that archaea have at least as much in common with eukaryotes as they do with bacteria. For example, archaea cell walls and membranes are chemically different from those of bacteria. The membranes of archaea contain lipids that are not found in any other type of organism on Earth. And bacteria have a polymer called peptoglycan in their cell walls, which archaea do not. The amount of peptidoglycan in their cell walls is an important characteristic of bacteria. Bacteria are often classified into one of two groups based on this difference, like is shown on the screen here. A staining method called a Gram stain is used to tell the two groups apart. The Gram stain is important for diagnosing infectious bacterial diseases, and it sometimes helps determine the type of medicine a doctor chooses to fight infection. Because of their cell wall differences, archaea are often not affected by medicine used to treat bacterial infection. Third main idea. Bacteria have various strategies for survival. The strategies that prokaryotes use to survive and to transfer genes have made them very successful at adapting quickly to almost any habitat. Prokaryotes reproduce by a type of cell division called binary fusion, which means division in half. While the chromosome copies itself, the cell grows. When the cell has about doubled in size, its plasma membrane grows inward and divides the cell into two equal-sized daughter cells. Each daughter cell will contain a complete copy of the parent cell's genes. Although prokaryotes reproduce asexually, they still have the ability to exchange genetic material in several ways. In one process, called conjugation, prokaryotes can exchange parts of their chromosome through a hollow bridge of pili formed by connecting two or more cells. During conditions unfavorable for survival, some bacteria can produce an endospore, a specialized cell with a thick protective wall. To form an endospore, the bacterium copies its chromosome and produces a wall around the copy. This thick wall around the bacterial DNA helps it to survive harsh conditions such as drying out, temperature change, and disinfectants. Endospores can last for centuries. Some have even been found in Egyptian mummies. This video has been Chapter 19.4, Bacteria and Archaea. Key concept: bacteria and archaea are both single-celled prokaryotes.